Is it okay for you to stand here, use the screen, then we can see what you're doing? So, yeah. Okay, check this out of the way.
Good afternoon, everyone. My name is Sabah, and I'm going to present or give an update on my project. The project is related to a biosensor for the detection of Lyme disease using different novel strategies. So you can see the title of my project here. So just give you a brief introduction, like it's about the detection of Lyme disease, particularly about the protein OSPC. It's the protein present in the outer surface of the bacteria. So I will quickly go to the main part of the project, that is my methods and materials. The first one is the synthesis of carbon quantum dots. I use 3 different methods to prepare the carbon quantum dots to see which one is more effective for related to its fluorescence and detection of my particular bacteria. The first method is I use citric acid and Aptes for the surface functionalization of carbon quantum dots. I used 0.5 gram of citric acid and 10 ml of Aptes, and then I put in hydrothermal reactor for 5 hours at 200 degrees centigrade. Then after that I filtered the solution using the 0.2 micrometer of filter paper, and then I checked the absorbance and fluorescence of the solution. The second method was using citric acid plus ethylene diamine and using DI water, and the procedure is same, but the timing I use is 4.5 hours at 180 degrees centigrade. Then the rest of procedure is same. Then put in hydrothermal reactor for the mentioned time, then filter in the solution, and at the end check the absorbance and the fluorescence. So here is the first solution, which is color yellow, and the second one is EDA. You can see the color difference clearly. Okay, the third method I use citric acid and urea. The first is in Aptes, the second I use citric acid and EDA. The third one I use urea for the nitrogen doping. So I put into hydrothermal reactor for 5 hours at a temperature 180 degrees centigrade. The rest of procedure is same. But here, first I use citric acid and urea, and after hydrothermal reactor, I added the Aptes, around 4 ml of Aptes for surface functionalization, and then put on the stirring for 2 hours, around 400 RPM at 60 degrees centigrade, then filter the solution and check the absorbance and fluorescence. Here is the end product. The color is dark green. So the results, the first result is showing the results for citric acid and Aptes. So the first graph you see here is the UV absorbance, which is around 300 to 350. The peak is at 340, and which is the higher as compared to the rest of the two methods. So the second graph is showing the fluorescence. I excite at different wavelengths. The first wavelength around 330, second is 340, and third is 350. And the fluorescence intensity of this one is not higher as compared to second one, because here I use urea, which is like the nitrogen doping, and also the surface functionalization using the Aptes. But here the intensity is lower as compared to the second one, because I didn't use urea here. So clearly see the difference in the fluorescence intensity and also the absorbance. But absorbance for the second method is lower as compared to the first one due to the composition of my material. So the third method, I didn't use urea and not use the Aptes. I used ethylene diamine for getting the more fluorescence. So here you can see that the graph for absorbance is the same, 300 to 350, like the urea one. And here the fluorescent intensity is good. It's consistent using by exciting a different wavelengths from 320, 330, 340, and 350. You can see the consistent in the maybe the size is good in this one, but the fluorescence intensity is lower as compared to the second method where I use the urea. Maybe due to the surface, because I just did UV absorbance and fluorescence. I have to check some results also in FTIR to see what's the functional changes occurred during the reaction. So these are the three results. So there are a lot of pending results. I'm still waiting to check the particle size, also the functional groups confirmation using the FTIR in the next week, and also for the surface morphology of all the three methods, what changes have occurred either in size, either in fluorescence or functional properties, because I use different chemicals like one of the method I use aptas, one of the method I use citric acid, one of the method I use urea. So it's very important to look the differences in the fluorescence and also the functional properties. So I'm going to use XRD to see the crystal structure and also TGA to see the thermal stability. And along with that, I am also going to continue with this project by preparing the molecular imprinted polymers to use along with the carbon quantum dots and to see the detection of OSPC protein in the bacteria. Also, I am going to run an experiment with NAMD using the copper, indium, selenium, zinc sulfide quantum dots and also to characterize them along with these experiments. So these are just updates and these are my upcoming tasks for the project.
Okay. All right. Great. Thank you so much. Yeah. Okay. Any questions? Suggestions? Or yeah, any questions? No? All right. Thank you so much, Sabah.